Good evening, everyone. So, yesterday we have stopped till coupling and repulsion linkages. Okay. Now we will discuss about types of linkage. So, here linkage is classified into different types based on the presence or absence of crossing over and what are the genes involved and the chromosomes involved. Okay. So, first is based on the crossing over. There is complete linkage and incomplete linkage. Okay. So what is complete linkage? So linkage in which crossing over does not occur is known as complete or absolute linkage. That means on a chromosome when different genes, so the position or the location where like we'll have address for our house. So in the same way, each gene will have a location and that location is called as locus. If it is a single gene, single location, it is called locus. Its plural is loci. So, if multiple genes are present on different loci, which are very close to each other, very nearby, so you cannot expect any crossing over between those genes. And so, definitely those genes will transmit together to the next individual progeny. So, such kind of linkage is called complete or absolute linkage. And in other words, when only parental types are obtained from the test cross progeny, it refers to the complete linkage. That means when there is no crossing over or recombination, you cannot find any new phenotypes or new characters. So only parental characters are found. Okay. So that is called as complete linkage. So example is Drosophila male and female. So here, where the parental combinations of characters appear together for two or more generations. Okay, regularly you can see same kind of pattern. This happens when a chromosome does not undergo any breakage by accident or during gametogenesis. And the genes for bent wings, so in Drosophila, and shaven bristle, which are present on the fourth chromosome of Drosophila, they exhibit complete linkage. Okay. And the almost complete absence of independent assortment between these two gene pairs is the evidence of very strong linkage. Okay. And all the genes of male Drosophila, they remain in complete linkage. Okay. That means especially these two gene pairs. So they actually inherit together. You cannot expect any independent assortment. So I told you what is independent assortment. When two genes, they segregate separately. Okay, there is no compulsion that if one allele comes, the other also, it has to pass the same allele, similar allele. So there is no such compulsion. Both independently assort. So such kind of scenario is not seen in this case. So here is the example, but it is blur. So you can see it from the textbook, like, okay. So the print is very small, so it is not possible. Okay, yeah, here you can see. So in the parent, they are homozygous for AB, okay, capital A, capital B, and that is two genes. A gene, so one parent is having capital A allele, other parent is having small a allele. And for capital B gene, for B gene, one parent is having capital B, other is having small b. Okay. So A, B, both are capitals, A, B, both are small. So the first generation, what happens? There will be hybrid. So one chromosome from this parent and one chromosome from that parent. Therefore, capital A, capital B, small b. A hybrid variety will be obtained. So when it is test crossed, that is, it is crossed with a recessive parent, that is A, small a, small a, small b, small b. What happens? This heterozygous or progeny will give four combinations: capital A, capital B, small a, small b, capital A, small b, small a, capital B. 4 combinations you'll get. But here, as we have small a, small a, small b, only you can expect small a, small b combination. Now, if we draw a Punnett square, so what kind of combinations you can expect? Capital A, capital B, small b in one. Capital small b from other. Capital A, small b in another. And small a, capital B, small b in another. So you have 1:1:1:1 ratio. This is independent assortment. That means you see every allele is separately passed to the next generation. There is no rule that if capital A comes, then only capital B has to come. Okay, no, all kind of combinations are possible here. Then we'll see complete linkage. So here you see same. One parent is homozygous for capital A and capital B, and other parent is homozygous for small a, small b. In F1 generation, they got heterozygous, capital A, capital B, small b, heterozygous progeny. When test crossed with homozygous recessive parent, they got, as there is a linkage, definitely when there is capital A, only capital B should come. And when there is small a, only small b should come. So here you can see only 2 combinations instead of 4. So when these 2 are combined with the recessive parent, you can see capital A, capital B, small b, small a, small b, small b. Although here 1:1 ratio, but here only 2 combinations you will get. Okay. Or the linkage can be of other type also. That is, if capital A, small b are linked and small a, capital B are linked. Okay, you don't know like which, this is example. Okay. In, there is no rule that if homozygous, that is dominant allele of one gene should be in linkage with the dominant allele of the other gene. So are you getting my point? That means if capital A should only link with capital B only, there is no such rule. Capital A can link with small b also. So in such case also, you will get 1:1 ratio, but the combination will differ. Okay. That is called complete dominance. Then you have incomplete dominance, where you can see 20% of recombination. So here, one combination is same, till test cross it is same. Now, here you can see capital A, capital B, small b, capital A, small b, small a, capital B, 4 combinations you got. But they are incomplete linkage, so they are not in equal ratio. That is 1:1:1:1, you cannot find. The ratios will differ. So always parental combinations will be more. So here you see 4:4:1:1 they have got. That means the dominant combination was more. So some are in higher ratio, some are in lower ratio. The proportions will vary. But you will get the combinations are similar. So there is another type also here. So this is called incomplete dominance. Sorry, incomplete linkage. Okay. Yeah. So now in incomplete linkage, if some frequency of crossing over also occurs between the linked genes, it is known as incomplete linkage. So when recombination occurs after a test cross, so even in the test cross progeny, besides parental combinations, if any recombination occurs, then that is called as incomplete linkage. So usually incomplete linkage occurs, and that was observed in maize, pea, Drosophila female. So complete dominance was in Drosophila male. Okay. And linked genes are located in chromosome and have chances of separation by crossing over. And this is called incompletely linked genes. And the phenomena of their inheritance is called incomplete linkage. So examples are pea, tomato, female Drosophila, mice, man, poultry, etc. So here you can see the examples at the top. So whatever I have discussed. Then second basis is based on the genes involved. So here it is complete linkage and repulsion linkage. So what is complete linkage? It refers to the linkage either between dominant genes or coupling linkage. Coupling linkage refers to the linkage between either dominant genes or between recessive genes. So this is observed in pea, maize and other crops. So there is no either both will be dominant or all are recessive, but no other way around. Okay. Then repulsion linkage is linkage of some dominant genes with some recessive genes. So here also some genes in pea, maize and other crops. Then third criteria is based on chromosomes involved. So here we have autosomal linkage and X chromosomal linkage. So autosomes are the chromosomes which are involved in the characters that are not what you call not sex chromosomes. Okay, so they are somatic chromosomes present in the somatic cells basically. So if refers to the linkage of such genes located on the autosomes, whereas X chromosome chromosomal linkage are the linkage of genes present on the sex chromosomes. You can see these chromosomes only in the gametes, but not in the somatic cells. Okay. Then linkage groups. So linkage group refers to a group of genes which are present on one chromosome, and the number of linkage groups is limited in each individual. Okay. The maximum number of linkage groups is equal to the haploid chromosome number of the organism. What is haploid? One set of chromosomes are called haploid, two sets of chromosomes are called diploid. So in the haploid chromosomes, how many linkage groups are there? That is the maximum number of linkage. In case of species having dissimilar sex chromosomes, so the linkage groups are one more than the haploid chromosomes. For example, there are 10 linkage groups in corn, 7 linkage groups in garden pea, 7 in barley, 4 in Drosophila melanogaster and 23 in Homo sapiens. So because the homologous chromosomes will be equal. So therefore that is if human beings are there. So 23 pairs of chromosomes. So 23 and other pair is also 23. Equal number of chromosomes. Therefore the groups also, the number of groups will be same, 23. If in some organism, suppose 23 chromosomes in one and other set will be some 24 or 25, in such case, the groups will alter. Is it clear? So genes are assigned to various chromosomes of a species with the help of deletion, monosomic and nullisomic analysis. And these linkage groups are assigned to different chromosomes in a linear fashion. Okay. And same sequence usually as they are present. Okay. And a relative length of various linkage groups in a species exhibit a close agreement with the relative length of chromosome in which they exist. Okay. That means the length of the groups is relative to the length of the chromosome in which they are present. Then detection of linkage. So how can we expect or detect a linkage? So test cross will do that. F1 progeny is crossed with the recessive parent. So test cross is the most common method of detecting the linkage. In this method, the F1 heterozygous at two loci is crossed with a double recessive parent and the phenotypic test cross is exhibited. If the phenotypic ratio is 1:1:1:1, having parental and recombinant genotypes, it indicates absence of linkage. But if the ratio is 1:1:1:1, but it deviates, that is parental types are only present. So not only the ratio also sometimes get deviated and only parental types are seen and the recombinant types are absent, then we can expect a linkage in such kind of genes. Then next is linkage and pleiotropic. A close association between two or more characters may result either due to linkage or pleiotropic or both. So what it is pleiotropic refers to the control of two or more characters by a single gene. So a single gene, it codes for different characters. So if that gene is present, obviously the person will be having all the two or three characters together. In such case, a tight pleiotropic effects also may be present, and a tight linkage between two loci can also be confused with a pleiotropic. And the only way to distinguish between the linkage and pleiotropic is to observe the crossover product between the linked characters. Okay? Intermating in segregating populations may break the tight linkage, but a huge population has to be raised to find out the crossover product. Okay, sometimes when the mating occurs, so the segregation may happen and the linkage may break in some generation. And if a crossing over product is not found in spite of repeated intermating, it is the case of pleiotropy rather than linkage. That means if the linkage is present, it might break after some generations. But if it is pleiotropy, it won't break because it is only single gene, right? So it won't break. It will code for different characters. So that will remain same after any number of generations. Then theories of linkage. So there are two theories which explain the phenomena of linkage. That is differential multiplication theory of linkage and chromosome theory of linkage. So what is differential multiplication theory of linkage? So this was given by Bateson in 1930. So where the segregation of characters during gamete formation, the set of gametes having parental combinations, they multiply more rapidly than the set having the non-parental combinations. As a result of this, great number of gametes and thus the offspring with parental combinations will be formed. So there is no cytological basis for this theory. However, so this was severely condemned because after segregation, only a single division occurs before the gametes are formed. So according to this theory, they said that the gametes with parental generations, they divide very rapidly, but that is not the case. Only a single division occurs. So whether it is parental ones or recombinant ones or linked ones or whatever it is, crossing over. So only one, what you call, one division occurs. So there is no cytological proof for that. Okay. Then second theory was chromosome theory of linkage. This was given by Morgan and Castle. So here, according to this, the genes which show the phenomenon of linkage are situated on the same chromosome, and these are linked genes usually remain bounded by the chromosome material and they cannot be separated by a process of inheritance. And the extent of genes is correlated with distance between the genes in the chromosome. So closer the genes, stronger the linkage, and farther the genes, the linkage strength will be less. So the genes are arranged in linear fashion in the chromosomes, and this was suggested by cyclogenesis, and this theory was accepted, and even today we follow this theory. Okay. So why we need to study the linkage and what is the significance? So it reduces the possibility of variability in gametes unless crossing over occurs. Then it plays important role in determining the nature of hybridization and selection programs. It reduces the chance of recombination of genes and they hold parental characteristics together. So therefore, it helps the organism to maintain its parental, racial and other characters. So in plant and animal breeders, so this is very important because when there is linkage, it will be difficult to find various characters. Okay. So this is about the linkage. The next topic is crossing over. So during meiosis breakdown, breakage of chromosomes occurs accompanied by interchange of corresponding parts. So now you have two sets of each chromosome. Chromosome 1, two pairs. One pair, okay, two chromosomes. Then chromosome 2, 2. Chromosome 3, 2. Like that. So the corresponding parts will be interchanged between chromatids of homologous chromosomes. So Morgan termed this phenomena as crossing over, and it is the process that produces new combination or recombination of genes by interchanging the corresponding segments between non-sister chromatids of homologous chromosomes. Okay, during meiotic phase, that is packaging stage. In other words, crossing over results from the exchange of genetic material between non-sister chromatids involving breakage and reunion of precise point. At a specific point, when there is a breakage occurs, it exchanges with the non-sister chromatid. The term crossing over was first used by Morgan and Cattell in 1912. Okay, so here there are some differences between the crossing over and linkage. So here, crossing over leads to the separation of linked genes, whereas in linkage, it keeps the genes together. And crossing over involves non-sister chromatids of homologous chromosomes, whereas linkage involves only individual chromosomes. There is not, a pair is not required. Then frequency of crossing over can never exceed 50%, whereas linked groups can never be more than haploid chromosome number. Then crossing over increases variability by forming new gene combinations, whereas because we can get new combinations, whereas in linkage it reduces variability. And crossing over provides equal frequency of parental and recombinant types in test cross, whereas in linkage, high frequency of parental types than the recombinant types are observed in the test cross progeny. So these are the main differences. So the crossing over provides high frequency of parental types than the recombinant types in the test cross progeny. So we'll have some features, some important points here. Crossing over takes place during packaging stage of meiosis, and each pair of chromosome has 4 chromatids at that time. And crossing over occurs between non-sister chromatids, and thus one chromatid from each of the 2 homologous chromosomes is involved. Because in this stage, the division of chromosomes, the replication occurs, but separation doesn't occur. So therefore, you have 2 pairs of homologous chromosomes. Therefore, the exchange occurs between the non-sister chromatid of homologous chromosomes. That means in one cell, you will have 2 sets of each chromosome. Suppose chromosome 1, a pair are there, 2 chromosomes. In another cell, again 2. So before the division, there are 4 sets of chromosome 1. So between non-sister chromatids of these chromosome 1 itself, for example, so the crossing over takes place. And crossing over occurs yeah, that's I told you. Okay, each crossing over involves only two of the four sister chromatids of two homologous chromosomes. However, double or multiple crossing over may involve four or three or two of the four chromatids, but it is very rare. And it is universally accepted theory of crossing over, which takes place at fourth strand stage. Okay. And crossing over leads to recombinations or new combinations between linker genes, giving recombinant types or crossover types and two parental or non-crossover types. And crossing over leads to exchange of equal segments or genes, and recombination is always reciprocal. Okay. However, unequal crossing overs also may be reported. And the value of crossing over or recombinants, it doesn't exceed 50%. So maximum it is 0% means no crossing over, 50% means 50% of the crossing. Okay. And the frequency of recombinants can be worked out from the test cross progeny, and it is expressed as the percentage ratio of recombinants to the total population. So here you can see crossing over frequency is given in the percentage, how much crossing over occurred. So number of recombinants divided by total progeny. So total means recombinant plus parental type. Okay, into 100. So cases of two-strand crossover, somatic crossover, sister strand crossing over, and unequal crossing over are also known. And the frequency of such cases is extremely low. So that is called infractions. And the crossing over differs from linkage in specific aspects. Okay, I told you what are the several aspects, how it is different. So the example here is a test cross between the F1 hybrid of grey body and long wings and a double recessive, that is test cross. So this results in the Drosophila having a black body and vestigial wings. Okay, so that means here F1 progeny is grey body, long wings, crossed with black body, vestigial wings. Then F2 generation, so they got grey body, long wings and black body, vestigial wings. That is equal number of parental. Then for the same characters, if the sex is reversed, if the female hybrid is backcrossed with the double recessive male, they got grey body, vestigial wings and black body, long wings. Reverse, they got reverse. A new combination was observed. So what is the mechanism of crossing over? So during the zygote stage of the first prophase of meiosis, these homologous chromosomes, they pair. This is called as dyad or two-strand stage. And during Paquetin stage, each chromosome becomes twisted into two chromatids. This is called four-strand stage, which is called tetrad. So it is at this stage the crossing over takes place. And the chromatids of four-strand stage, that is tetrad, get spirally twinned, forming one or more points of contact, which is called chiasma. And at chiasma, only two chromatids participate in the chiasma, and each of these two chromatids, they belongs to different number of homologous pair of chromosomes. So during this diplotin stage, the chromatids at chiasma, they break and they rejoin and they form again homologous parts are interchanged. And here the new chromatids acquire a changed genetic constitution. This is called as crossing over. Okay. And what is the frequency and factors affecting the frequency? So the frequency with which the crossing over occurs between two genes is called frequency of crossing over. And here, as I told you, it is expressed in percentage. Generally, it is directly proportional to the distance between the two genes. If the distance is high, the percentage will be more. Means if they are nearby, linkage chances are more. If they are far, then crossing over chance is more. And vice versa. Thus, the percentage of crossing over between two genes indicate the distance between the two genes. Okay, so this is very important for constructing chromosomal maps of animals and plants. So here there are various factors which control the frequency of crossing over. Plow showed that the high and low temperature increases the frequency. So temperature. Then Hansen, these are scientists, okay? They showed that their radiation by radium increase frequency. Then gene mutations also decrease the frequency. Chiasma formation at one point discourages the chiasma formation in another point which is near vicinity. That is called interference, which decreases the frequency. And the interventions of chromosomes are also known as suppress crossing over. And all these factors, they affect crossing over. They also affect the linkage. That means if the crossing over is decreased, then the chance of linkage will be more. Inversely proportional. Okay, the relation is inverse. The next is chiasma and crossing over. So the point of exchange of segments between non-sister chromatids during meiotic phase, that is chiasma, and it is thought to be the place where crossing over takes place. And depending on the position, chiasma is of two types. Terminal means at one end and interstitial. When the chiasma is located at the end of the pairing chromosome, it is called terminal. When it is located in the middle part, it is called as interstitial chiasma. And later, this interstitial chiasma is changed to terminal position by the process of chiasma terminalization. And the number of chiasma per bivalent chromosomes may vary from one or more depending on the length of the chromosomes. When two chiasmata are formed, they involve 2, 3 and all of the chromatids. Okay. Then what is chiasma terminalization? So the movement of chiasma from middle part to the terminal is called terminalization. And the total number of chiasmata can be terminalized by, it depends on different stage or time. So these are the factors that affects the terminalization. So usually this terminalization occurs between diplotin and metaphase 1. And there are three theories that explain this terminalization. One is electrostatic hypothesis, other is coiling hypothesis, and third is electrostatic chromosome repulsion theory. So in the electrostatic hypothesis, terminalization takes place due to localized repulsion forces in centromere and generalized repulsion forces on the chromosome surface during diplotin stage. This is called electrostatic hypothesis. Then coiling hypothesis. According to this hypothesis, terminalization takes place by mechanical tension developed in the chromosome due to coiling. And this tension force becomes greater than the force binding the chromatins. Therefore, the terminalization takes place. And the other one is elastic chromosome repulsion. According to this, all bodies having the definite shape, they resist any kind of change that leads to alter their shape. So this chiasma forces the chromosome out of its shape by binding force, which leads to the development of repulsion at the point of exchange, resulting in the terminalization. The next is interference. So here Muller, he coined the term interference, where the tendency of one crossing over to reduce the chance of another crossing over in the adjacent region. So interference is affected by the gene distance on the chromosome, and lesser the gene distance, greater the interference, and vice versa. And generally, it is observed that crossing over in one region of chromosome may check or may stop the crossing over in some second region. Okay. So sometimes the presence of recombination in one region enhances the chance of recombination in adjacent region, and this is called as negative interference. And this type of interference is seen in Aspergillus and bacteriophages. And this coefficient of interference can be estimated by interference minus coefficient of coincidence into 100. This is the formula by which we can calculate. So what are the difference between the positive and negative interference? Positive interference, where one crossover reduces the chance of another crossover of adjacent region, observed in both eukaryotes and prokaryotes, and coefficient of coincidence is less than 1. Sorry, this is, I think, 1. Sorry. Yeah. So the negative interference is one crossover enhances the chance of crossing over in adjacent region, and it is observed in some lower organisms, and the coefficient of coincidence is always more than 1. So the coincidence is, actually this is coined by Muller. So here the coincidence explains the strength or degree of interference. So the coefficient of coincidence is the percentage ratio of observed double crosses to the expected double crosses, and the greater the coincidence, lesser will be the interference. So if coincidence is more, interference will be less. If interference is more, coincidence will be less, like that. So coefficient of coincidence is observed double crosses divided by expected double cross. So the coefficient of coincidence is a measure of intensity of interference because it has negative association with interference. So the value of coefficient of coincidence is less than 1 for positive interference and 1 for absence of interference and 0 for complete or absolute interference. Then chromosome mapping. So chromosome map is a linear diagram where it depicts the locations of various genes on the chromosome and recombinant frequency. Such type of maps are called as genetic maps or linkage maps. And the process of assigning genes on the chromosome is known as chromosomal mapping. And this is done by the 3-point test cross. A 3-point test cross is a cross of F1 generation with homozygous recessive parent. So as we were calculating previously, and the 3-point test cross, it gives useful information about the sequence of the genes, in which sequence the genes are present, about the recombination frequencies between genes, and this is essential for mapping of chromosomes. So how the gene sequence is observed? So it is determined by the crossing over between the two genes. Okay. So the greater number of recombinant percentage between the two genes, there is the more distance is there. Suppose between A and B, if it is 19.92 and 21.75, they are nearby. So the crossing over percentage is less. Whereas between A and C, the distance is more. So the crossing over will be more. Okay. And then the coefficient of coincidence, which is observed double crosses by expected double crosses, and the interference, that is 1 minus coefficient of coincidence. So these are observed by the test cross when performed. So based on the number of organisms observed, based on the ratios, we can actually give the spacing. Okay. So if you see small a, small, oh, sorry. Yeah. So here, capital A, small b, small c are there. So when after crossing and everything, test cross was performed, they got 1,000 progeny. So there were some spelling mistakes here. You have to just check it. So after 1,000 progeny, small a plus, small b plus, small c plus. Okay. So the combination is parental genotype. So it is 244. So majority, so there is no recombination. Then A and B are there, but C. So C is not there. So this is because a recombination between capital B and capital C. So here 81 are there. So recombination occurred. Like that, they will check different loci. Okay. Then here, so there are, if you see here, so total of 350 progeny were showing out of 1,000. 350 were showing recombination between A and B. 220 were showing recombination between B and C. Now how this is double cross. So how to check the double combination? 350 divided by 1,000 into 220 divided by 1,000. So this gives 77 individuals per 1,000. Okay. But there are actually only 50 double recombinants. But expected is 77. So observed is 50 divided by 77, which is expected. So this gives 0.65, which is the coefficient of coincidence. And 1 minus coefficient of coincidence gives the interference. That is 0.35 will give the interference. Okay. Then types of crossing overs. So depending on the number of chiasmata formed, there are three types. Single crossover, double crossover and multiple crossover. In single crossover, if only one chiasmata is formed, chiasma, then single crossover. It involves only 2 chromatids out of 4. Then in the double crossover, if 2 chiasmata are involved, then here we can observe double crossover. And 2 or 3 of 4 strands will be involved and the ratio will change. Okay. Then multiple crossover is more than 2 chiasmata are present. Then it is called multiple crossover and it can be triple, quadruple and so on. The frequency is very low or rare. So what are the factors affecting the crossing over? So the frequency of crossing over is influenced by distance between the genes. If the distance is more crossing over. If distance is less crossing over and more chance of linkage. Then age. Generally, crossing over decreases with advancement of age in the female. Okay. Then temperature, especially in Drosophila. Okay. So the rate of crossing over in Drosophila increases above and below the temperature of 22 degrees centigrade. Then sex. The rate of crossing over also differs with the sex. That is, in male, there is complete lack of crossing over. In male Drosophila and female silk moth. Okay. Then nutrition. Presence of metallic ions like calcium, magnesium in food can reduce the recombination in Drosophila. And removal of such chemicals from the diet increases the rate of crossing over. Okay, this is all in animal experimentations. Now, chemicals. Treatment with mutagenic chemicals like alkaline agents also increases the frequency of crossing over. Then irradiation. Irradiation with X-rays and gamma rays also enhance the frequency of crossing over. Structural changes. due to inversions and translocations of chromosomal segments also affect the crossing over. Then centromere effect. Generally, genes that are located adjacent to the centromere, they show reduced percentage of crossing. And cytoplasmic genes. So cytoplasmic genes lead to the reduced crossing over. For example, Tifton male sterile cytoplasm in pearl millet. Okay. So why we need to study this crossing over? What is the significance? So it gives an evidence of that chromosomes are arranged in a linear fashion, and also to help to construct the chromosomal maps, which is very important for genetics to study the linkage in the organisms. And the phenomena is more useful in organisms because new combination of genes and variation in offsprings can be observed, and essential to study evolution of new species. And it is useful in three principles. One is creation of variability, locating genes on the chromosomes, and preparing linkage maps. So creation of variability is nothing but it leads to recombination of new combinations. So through which actually variation between different organisms was developed. Then locating genes. It is a useful tool for locating genes on the chromosomes, the order of genes and the location or loci. And the linkage maps. This plays an important role in preparation of chromosomal or linkage maps. Therefore, to prepare linkage maps, this is very useful. So this is the significance of crossing over. Okay. Okay, fine. So we'll stop here, and tomorrow we will continue with the sex determination. Okay. Any doubts here? Okay, if no doubts, let's end the class. Okay. Thank you.